Hello everyone, thank you for tuning in today. My name is Jaime Cordova. I'm a Solar System Ambassador on behalf of NASA and the Jet Propulsion Laboratory. I'm also a PhD candidate at the University of Wisconsin-Madison in the Department of Genetics. And today it's my pleasure to be here at UW Space Place to be talking to you about planetary protection and to talk a little bit about if we're ready for the search for life elsewhere. Now, this talk was originally motivated by the COVID-19 pandemic in March. The reason behind this is the SARS-CoV-2 virus is a new virus that we are still learning about 10 months later. And really, right around the time that the pandemic was starting, I started to think about, well, you know, we don't have any immunity to this virus now. What if there was ever the chance of life coming from somewhere else, and we had to deal with that? How are we going to deal with a virus that's coming from another world? Interestingly enough, there is a very false hypothesis, and I'd really like to stress how false this hypothesis is that the coronavirus, or SARS-CoV-2, was originally came from space. Now, this paper by Steele et al. Came out in July of this year, and essentially their hypothesis is that in October 11th of 2019, a meteorite landed in about 2,000 kilometers from Wuhan, China, the epicenter of the pandemic, and basically in their paper, they state that this meteorite was likely a fragile and loosely held carbonaceous meteorite that was carrying a cargo of trillions of viruses or bacteria and other primary source cells. Now, again, this hypothesis is very false. We somewhat know the origins of the SARS-CoV-2, but nevertheless, this is something that we do think about. So far, it's for a long time, it is a thing of science fiction, with movies coming out throughout the years. For example, the Andromeda strain in the 1960s, which is really the one that has focused on a possible microbe coming from another world or from another galaxy and infecting humans here on Earth and wreaking havoc. You also have, for example, extraterrestrials that are in the form of monsters, like in the movie Alien. Or you have extraterrestrials in a form of life that we don't really even understand. For example, in the movie Life that came out a couple of years ago. Now, while this has been a thing of science fiction for some time, it's still definitely something that's of realistic concern for us here on Earth. And this is an area that's known as planetary protection. Now, what is planetary protection? This is a practice of protecting the solar system bodies from contamination. There are two areas of planetary protection. One is backwards contamination, which is essentially preventing the contamination of Earth from returned samples. So, for example, during the Apollo missions, ensuring that those samples weren't contaminated by some form of possible lunar life. Then you have forward contamination, which is minimizing contamination of other worlds by Earth life. So essentially, when we're sending out rovers or when we're sending out probes to other worlds in the solar system, we want to ensure that they're carrying the minimal amount of life possible to ensure that we're not contaminating that world. Now, planetary protection had its beginnings in 1956 when the 7th Congress of the International Astronautical Federation in Rome held its 7th Congress, and so the planetary protection has been around for about a little bit over 60 years. The primary focus of this Congress was having conversations about a potential artificial unmanned satellite that would be taking off soon. And because that conversation was taking place about potential unmanned satellites taking off soon, there were also conversations regarding possible lunar and planetary contamination. Now, I'm sure no coincidence, the very next year in 1957, the Soviet Union launched Sputnik 1 on October 4th, and Sputnik 2 on November 2nd. Now, this was of great concern for scientists, not only because now the Soviet Union had made it to space, and in fact, we were the first ones to make it to space, but now it was really becoming a reality that we could potentially contaminate other worlds, especially now that we were actually launching things into space. Now, the very next year, in 1957, and actually I should backtrack, planetary protection to a certain extent has its beginnings here at UW-Madison. In fact, in my home department, in the Department of Genetics, where Professor Joshua Lederberg, professor of genetics and the head of the department at the time, he was a big planetary protection advocate, and he also had a lot of interest in exobiology, which now is known as astrobiology. And here he's pictured at his lab here at UW-Madison. Now, in late 1957, shortly after the launch of Sputnik 1 and Sputnik 2, Lederberg wrote to the National Academy of Sciences and multiple members of the National Academy of Sciences regarding concerns about the contamination of other worlds. And in fact, he sent out a memo that was, for the most part, privately shared, but he sent out this memo to several members of the National Academy of Sciences, and including other people who would eventually be leaders at NASA. And in general, his concerns can really be summed up by this first sentence in his memo known as Cosmic Microbiology, which says, quote, "I am deeply concerned over the incipient despoilment of an opportunity for biological exploration unique in our time, the starting point of extraterrestrial microbiology." So he had a lot of interest in not only exploring life elsewhere in the world, life elsewhere in the solar system, but more importantly, ensuring that we weren't going to be contaminating these other worlds. In February of 1958, after Lederberg had sent out these letters to multiple National Academy of Science members, the National Academy of Sciences urged the International Committee of Scientific Unions to establish the Committee on Contamination by Extraterrestrial Exploration, also known as CTEX. Now, at the urgency, essentially, this was the first formal statement regarding planetary protection that was ever really developed. And essentially, what the National Academy of Sciences resolution stated was, quote, "Biological or radioactive contamination of extraterrestrial objects could easily occur unless initial space activities be carefully planned and conducted with extreme care. The Council of the National Academy of Sciences of the United States of America urges the International Council of Scientific Unions to encourage and assist the evaluation of possibilities of such contamination and the development of means for its prevention." And again, this was the first time in February of 1958 that a formal statement had been released regarding planetary protection. 1958 was a big year for planetary protection, likely because of the very recent launches of Sputnik 1 and Sputnik 2 in the year before. June 1958, the National Academy of Sciences established the Space Sciences Board, which is still active today. And essentially what the Space Sciences Board does is it provides advice on all interplanetary contamination issues, amongst other things. That very same year, in October, NASA was established. So there was more formal exploration of space. The same month, in October 1958, by no sheer coincidence, the Committee on Space Research, or COSPAR, was established. And essentially what this COSPAR is still active today, and the scope of COSPAR includes spacecraft sterilization and planetary quarantine. And this is actually still one of the main roles of COSPAR, is that they are in charge, or they are where the committee that's involved primarily with planetary protection today. CTEX, the original committee that I mentioned earlier, was absorbed by COSPAR. And COSPAR was an international effort that included the then Soviet Union. And this was a really big deal to ensure to include the Soviet Union as one of the basically leaders in this group. The Soviet Union and the U.S. examined approaches to avoid the transfer of terrestrial organisms to other worlds, now that the two leaders of the world were likely going to be launching into space soon, or had already launched, or were going to be launching into space soon. December 1958, another committee was formed. The United Nations developed the Committee on the Peaceful Uses of Outer Space, and this would eventually go on to form the United Nations Space Treaty, which I'll talk about shortly. September 1959, the Space Sciences Board made the first recommendation to NASA to develop sterilization techniques. These were implemented, or the reason that they wanted them to develop these sterilization techniques or procedures was so they could be implemented for spacecraft that would be passing near or eventually impact the moon. These sterilization techniques were being coordinated by the Jet Propulsion Laboratory, Goddard Space Flight Center, and also the Space Technology Laboratories. Now, the goal of that was to ensure that any spacecraft that would be passing near or actually impacting the moon wouldn't contaminate it. By pure, I guess, coincidence, you could say, that very same month, Luna 2 from the Soviet Union impacted the moon, and this was the first time that that anything had ever impacted anything had ever impacted the moon, and second, the first time that humans had ever made it to another world. Now there was a lot of this. The impact of Luna 2 on the moon caused a lot of great concern because the Soviet Union was shrouded to a certain extent in some secrecy, that it wasn't necessarily clear how sterile this probe had been. Now, Soviet scientists did assure scientists throughout the world that it was sterilized and that they did go through sterilization techniques, but exactly what those sterilization techniques wasn't necessarily clear at the time. And again, this caused a lot of concern for planetary protection advocates, such as Dr. Lederberg. From 1959 to 1962, the United States and the Soviet Union and COSPAR published guidelines. A couple of years in 1963, NASA acquired its first planetary quarantine officer, Captain Lawrence B. Hall, and the planetary quarantine officer would eventually become to what is now known as a planetary protection officer, who's in charge of these planetary protection guidelines throughout NASA, and in collaboration with other agencies throughout the world. Now, this planetary quarantine office had multiple contractors that were doing a variety of planetary protection research. And you'll notice the date of this was 1963. So at this point, 2 years earlier, President Kennedy had already challenged the nation to have a man on the moon by the end of the decade. So the work that was being done for planetary protection was critical for the Apollo program, particularly one with the return of lunar samples that would eventually come back to Earth, and two, the protection of the moon. So since astronauts were going to be landing on the moon and walking around, we wanted to ensure that despite there being a very slim chance that life could be on the moon, we wanted to ensure that we were protecting the moon and to not contaminate it. 1967, probably one of the largest formal agreements in regards to planetary protection was signed, which was the United Nations Outer Space Treaty that included articles on planetary protection. In particular, Article 9, which was again signed in 1967, that states, "Parties to the treaty shall pursue studies of outer space, including the moon and other celestial bodies, and conduct exploration of them so as to avoid their harmful contamination and also adverse changes in the environment of the Earth resulting from the introduction of extraterrestrial matter, and, when necessary, shall adopt appropriate measures for this purpose." So by now, this had become really a formal statement for any member of the United Nations to ensure that not only were we not contaminating another planetary body, but at this point, we were 2 years away from going to the moon, to ensure that we were not bringing anything back or being as careful as possible when we were returning something back to Earth. Now, because for this first part, I'm really focusing on forward contamination, I'm going to fast forward to the 1970s, mid-1970s, with the Viking missions. Now, by then, the Viking missions launched in 1975 and arrived at Mars in 1976. By this time, we had already made it to the moon and back, several times actually. And this was the first time that that NASA and really any agency was going to plan to get to Mars. Now, because at the time we really had an understanding that Mars, or we believed that Mars could be habitable or had the right conditions for life to be there, we wanted to ensure that we reduce the amount of contamination as much as possible. Now, what happened was NASA took extreme sterilization techniques, where essentially the Viking landers were built using material that could withstand high heat, because they were baked for 40 hours at 111 degrees Celsius. So to ensure to kill off as many possible microbes as possible. Yeah, this was built specifically for the Viking landers to ensure that we weren't contaminating anything on Mars. Now, after the Viking missions had landed and essentially showed us that that Mars wasn't as habitable as we thought, some of these restrictions were actually relaxed a little bit. So, for example, now some of the techniques that we have now, they don't necessarily all require, if we're launching to Mars, they don't all require, for example, that sterilization technique of undergoing baking for a very long time. It all really depends if it's going to an area that's of particular interest to astrobiology. Now, I'll fast forward to today with the current categories that we have for forward contamination. So NASA, or actually I should say COSPAR, has different categories depending on the planetary body and depending on the type of mission that basically rules to what levels the sterilization techniques for planetary production have to take place. So for Category 1 and Category 1L, these are bodies that are not of direct interest for the understanding of the process of chemical evolution or the origin of life. And this is really for any type of mission. Then you have Category 2 and Category 2L. This is for bodies that are of significant interest relative to the process of chemical evolution and the origin of life. But if we were to necessarily go there, the risk of contamination likely wouldn't compromise future investigations. So essentially we're saying it's critical for understanding the origin of life and chemical evolution, but odds are is that there isn't necessarily life there. Then you've got Category 3, which is specific for a flyby or an orbiter, and this is for bodies that are of significant interest to the process of chemical evolution and or the origin of life, and where scientific opinion provides a significant chance that contamination could compromise future investigations. So essentially what we're saying is this is a place that we are interested for chemical evolution and or the origin of life, and it also has a pretty good chance that life could have arisen there. Again, that's not to say that life did arise there, but the conditions are pretty good. And this type of planetary body applies to two different categories. So Category 3, which is for a flyby or an orbiter, and then also for Category 4, which is for a lander or a probe. Now you'll notice that this has two asterisks behind next to it, and this is what I was mentioning in regards to the Viking missions, or to the different categories that were made after the Viking missions, where depending on the type of area that we're going to on Mars, if it's an area that's particularly of interest to astrobiology, they have to go through extra steps to ensure that no contamination will take place. For example, they still undergo the same process of getting baked at 111 degrees Celsius, but now it's for 144 hours, so several days, almost a little bit over a week actually. And as this is in addition to other sterilization techniques that I'll mention shortly. Now, some mission category examples, for example, first for a mission or for a Category 1L, this is the moon. So this is because it's not of direct interest for understanding the process of chemical evolution or the origin of life. So by now we know that it's not necessarily, there likely isn't life there, just because of how dead the moon is. For Category 2, this is for bodies that are of significant interest relative to chemical evolution or the origin of life. So this applies to Venus, for example. And then also for that same Category 2 also applies to historic sites. So for example, we want to ensure not to contaminate any of the sites of the Apollo landings. Also for Category 2, this is for bodies that are, so another example of Category 2 is, for example, Titan. So we're interested in understanding the chemical evolution or the possible origin of life at Titan. But odds are is that if future missions won't necessarily contaminate that world. Then we've got Category 3, for example, which includes Mars, because this is a place where there's only a remote chance this is a place where, there's a place where we are interested both for the chemical evolution and the origin of life, and potentially future missions could contaminate that world. And then you've got, this is, again, Category 3 is focused specifically for flybys or orbiters, as opposed to Category 4, which has the same planetary body type for, again, Mars. So here I'm showing the Mars Curiosity rover. But in this case, we're focused specifically on where these rovers or where these landers are actually landing. Then finally, Category 5, which I'll talk about in detail a little bit later. These is dealing with sample return. So a restricted Category 5 restricted category is an area where we're returning samples from again, there is a very good chance that, the conditions are right for life to be there. For example, this would be the moon of Jupiter, Europa. Then there's also Category 5 that's unrestricted. which is, for example, the asteroid Bennu. The asteroid is dead. Odds are is that there isn't necessarily life there. And so the return of these samples is unrestricted, meaning that we don't necessarily have to take as great precautions as we normally would with, for example, something coming from Europa or Mars. Now, there are different sterilization techniques that are used currently. For example, any spacecraft that's being built is built in a clean room. This is a clean room of the Jet Propulsion Laboratory, and in fact, what's shown there is a diagram, or a mock-up of the Juno mission that's currently around Jupiter. So these are built in a clean room where the air is filtered. There's airlocks in there to ensure that nothing is getting in, and also that basically nothing is getting out. In addition to that, while the technicians and people who are building these rovers or these orbiters are in there, they actually also have to wear basically these full body suits, which are interestingly called bunny suits because you look like a bunny. And in addition to that, in addition to working in these airlocks, working into these large clean rooms, working in a bunny suit. Essentially, it's almost like second nature for people. If you're bored, or I read an article that said, if you're bored and you're just hanging out in there, what you're going to do is you're going to grab a rag, or not a rag, but a microfiber cloth, and clean up the spacecraft with either isopropyl alcohol or ethanol. Just to name two examples of different cleaners. So there are different sterilization techniques again, and this is in addition to occasionally getting baked depending on what area of Mars we're going to. Interestingly enough, with this isopropyl alcohol and the ethyl alcohol and the other cleaners that are used in these clean rooms, there are microorganisms that actually eat these cleaners. Now, that's not necessarily to they do swabs of the different orbiters or of the landers to check if there is microorganisms on there, and it turns out that there are. Now these microorganisms, there's a diverse amount. In fact, going into I believe about 300 or so, depending on the type of mission, but they are in low abundance. And in fact, that is one of the sterilization, or I shouldn't say sterilization techniques, but one of the requirements or characteristics of these missions. NASA knows and that it's very much near impossible to kill every single microbe that's on there. So they do have certain limits for how many microbes or how many cells, whether they be alive or whether they be dormant, can be on there. And in fact, the number is relatively low, so low that it's less cells than can actually fit on the lens of your camera phone. So that's that many cells that are spread out through a lander. For example, Curiosity, which is about the size of a car. You're really decreasing the chance that life could be traveling with these landers. Now, there are other protection methods, for example, with Cassini and with Juno. So with Cassini in 2017 was its end of mission. Because around Saturn you have the moon Enceladus, which is an area or a moon that has a lot of has a warm liquid ocean, or is believed to have a warm liquid ocean. We wanted to make sure not to contaminate it. So because Cassini was running out of fuel, and they wanted to make sure that it didn't lose control and end up crashing into Enceladus, then what NASA did a controlled burn into the atmosphere of Saturn, and it disintegrated in there, as opposed to end up just sort of essentially running off and crashing into Enceladus. And the same thing is going to happen with Juno, which is currently orbiting around Jupiter. So to ensure that it doesn't lose control and end up crashing into Europa, which is another moon that we think could potentially host life because it has a warm liquid ocean. It will eventually, at the end of mission, will crash or burn up, I should say, into Jupiter's atmosphere. Now, that was all really in regards to forward contamination. So in regards to how we're preventing the contamination of other worlds from probes or orbiters that we're sending from here on Earth. The other side of planetary protection is backward contamination. So this deals with the sample returns from other worlds. So there are two categories, as I mentioned earlier. There's unrestricted, which this is Earth return mission from bodies that are deemed by scientific opinion to have no indigenous life forms. For example, an asteroid. If we were to when OSIRIS-REx drops off its sample from the asteroid Bennu, that's not necessarily something of concern because it's an asteroid. The odds are is that there's no life there. That's as opposed to a Category 5 mission, which is restricted, where this is from our Earth return missions from bodies that are deemed by scientific opinion to be of significant interest to the process of chemical evolution and or the origin of life. So again, this could be, for example, Mars or Europa or Enceladus. Now, going back to the 1950s, or I should say to the 1960s, 1956, as I mentioned earlier, was the 7th Congress of the International Astronautical Federation in Rome. Just a couple of short years later, so very quickly after the planetary protection was first brought up, President Kennedy challenged to send a man to the moon. Now, because this challenge was now taking place, as I mentioned earlier, planetary protection advocates were very concerned about whether we were going to be contaminating the moon or whether we were going to be bringing life forms back from the moon, despite now it was a very, very small belief that there could be life there because we understood the moon enough to think that odds are is life couldn't exist there, but it was still something that was of concern. From 1962 to 1963, the concerns of scientists, the Space Science Board, Congress, and even commercial partners, they were all really ignored by NASA due to the chance of back contamination being, quote, too remote to warrant further attention. So essentially, again, these planetary protection advocates were saying, odds are is that there isn't any life there, or the chances are really slim, but we should still take it of concern. But even that very slim chance of concern, NASA wasn't necessarily concerned with dealing with that. In 1964, the Space Science Board hosted the potential hazards of back contamination from the planet's conference. And at this point, so despite NASA not necessarily sharing those same concerns about returning microorganisms from the moon or contaminating the moon, these planetary protection advocates continued on with developing either techniques, or I should say techniques or recommendations. And from this conference, the Space Science Board had some recommendations, including anything returning from the moon should immediately be placed in isolation. Astronauts should be quarantined for at least 3 weeks. And spacecraft, the suits, and the equipment should not be contaminated until thorough biological studies have been conducted on them. So essentially making sure that before we clean these off, is there anything on here that could be of interest or anything that could be life, and we want to make sure to catch it there before we wipe it down. And then finally, in regards to sample return, any preliminary sample analysis should be conducted behind biological barriers and under rigid bacterial and chemical isolation. Now, because this was all taking place, and I should say very quickly, I mean, this is 1964, 5 years later, Apollo 11 was launching and returning from the moon. Between 1964 and 1966, there was extensive planning, proposals, and discussions regarding the Lunar Receiving Laboratory, which was to be built at what's now known as the Johnson Space Center in Houston. In fact, there was so many conversations around this that they even risked losing congressional support because by this time, or really the original motivations behind Apollo and behind having all these, or this rush, I guess I should say, to get to the moon, was really all because of this fear of the Soviets. Well, by this time, there wasn't necessarily as much of the fear of the Soviets getting to the moon first, that Congress wasn't necessarily as scared to get astronauts to the moon and to return samples back. But essentially what this process consisted of was, as you can see on the picture on the right, astronauts would land, would splash down in the ocean, they would go into a mobile quarantine facility, and then they would stay aboard the aircraft carrier and be transported over to an island where there's runways. And that mobile quarantine facility would be placed into a cargo aircraft and then taken back to Houston. But before that was, or at the same time that was taking place, and a lot quicker, the lunar samples would be basically placed on a plane from the aircraft carrier and taken directly or as quickly as possible to Houston to the Lunar Receiving Laboratory. And the reason behind that was because they wanted those samples to get there as quickly as possible in case there was some form of, for example, radiation or some sort of outgassing that, because it had already been removed from the lunar surface, would degrade as time progressed. So the less amount of time that those weren't being studied, the better. Now for Apollo 11, this was obviously the first time that astronauts landed on the moon. There were these processes were followed. So, for example, on the left, I have an image of the splashdown shortly after they made it back from the moon. And what you can see here is these astronauts are wearing they're called BIGs, so a biological isolation garment. Interestingly enough, the diver from the Navy who went down there to get them out, he was wearing one himself. And he's actually from Wisconsin. I'm forgetting his name, but he's actually from Wisconsin. He was wearing one himself. He opened up the hatch. He tossed the 3 in there and closed the hatch back up. And the astronauts had to put on those garments inside. They put those garments inside, and then they walked out. They jumped into this little, basically like a dinghy, and then they were washed down with bleach to ensure that they weren't carrying anything on them. They were transported to the helicopter and then put into this mobile quarantine facility where they stayed for, they stayed on board the aircraft carrier, the USS Hornet, until they made it back to land, and then were transported by cargo ship. And then they could finally walk out once they were in the Lunar Receiving Laboratory, where they stayed in quarantine there for several more weeks. Interestingly enough, while they were on the USS Hornet and inside the mobile quarantine facility, President Nixon visited them. And NASA was taking such great precaution, and I should say that eventually they did take this a lot more seriously. NASA took such great precaution with ensuring that there was no contamination, that as they were walking over to the mobile quarantine facility from the helicopter, everything was being wiped down. Everywhere where they were walking was being wiped down. And once they were put in the mobile quarantine facility, they could take off those suits. But just in case there was a helicopter waiting the whole time for President Nixon, should some form of biological leak take place, that he can be evacuated off. And for example, at that point, the USS Hornet would have to be placed under quarantine or placed in isolation to ensure that nobody got sick. Eventually, they were released from quarantine on August 10th, 1969. Now, there was a lot of concern because they walked out at, I believe it was 9 p.m. of that night. And they were originally supposed to head out August 11th at 1 a.m. to prevent any press from talking to them or bugging them. But they were desperate to get out of quarantine and see their families, that they walked out at 9 p.m. Because they had already been deemed to be safe, talked to the press, and then were rushed home to see their families. This is after, again, 21 days that they had been in quarantine. Now, in regards to the samples, so the samples, as I mentioned earlier, were very quickly flown from the aircraft carrier to Houston because, again, they wanted to ensure that they were as pristine as possible, I guess you could say, because by this point they had already been traveling from the moon for a couple of days. So to prevent any radiation or any sort of chemical outgassing, and not necessarily be able to understand that scientifically. So these were rushed over to the Lunar Receiving Laboratory, and there they actually had to go through several sterilization techniques. So they underwent, the tubes themselves underwent UV light exposure. They were cleaned by biocides that were typically used for tanks or pipelines that were in contact with food or water. And then they were also placed into these airlocks to ensure that nothing was basically outgassing from them, and until they finally went into these glove boxes that you could see in this image on the right. And there was negative pressure. There was air being that's filtered throughout the Lunar Receiving Laboratory, and there was negative pressure within the Lunar Receiving Laboratory to ensure that nothing was getting out. So there were a lot of precautions that were taking place. Interestingly enough, one of the other something that I just found out while researching for this presentation is that the lunar material was also tested to check if there were life forms that were capable of generating disease or reproducing in certain types of organisms. And they did this by putting either, for example, mice or Japanese quail or cockroaches or moths or including, I believe, over 30 types of plants that were important economically. They put them all near the lunar samples to ensure that they wouldn't die, to ensure that there was nothing that's basically jumping off from these lunar samples and onto these different organisms and killing them off. And these samples had to stay under quarantine, or I guess I should say under isolation, for months on end before they can actually be passed on to different laboratories throughout the United States. Now, this was for Apollo. As I mentioned, there are missions that are currently taking place, or that have taken place, I should say. So Apollo 11, Apollo 12, and Apollo 14, those were all Category 5 restricted missions. After Apollo 14 and the realization that likely there wasn't necessarily anything on the moon that could really harm humans or any other terrestrial organisms, after that, those planetary protection techniques were sort of relaxed, at least in regards to, for example, they didn't have to wear those big suits anymore. Now it was just sort of a mask to prevent any potential bacteria from being exhaled. Now, going towards now to the focus of sample return missions, there are Category 5 unrestricted missions that have taken place. So, for example, this is the mission Stardust that returned a sample in 2006. It collected samples from a comet. Those samples were dropped off on a flyby, and those samples were collected and further studied. You also have Hayabusa, which in 2010 delivered a sample return mission from an asteroid, and this was from the Japanese Space Agency. Again, this is a Category 5 unrestricted mission because comets and asteroids, we don't believe that there's something that anything necessarily there could really harm humans. Then you have Hayabusa 2, which in fact is going to be making its tap and go essentially to collect some samples in the next coming days, I believe on October 20th. And then OSIRIS-REx, which will be returning its sample from the asteroid Bennu in 2023. And again, these are unrestricted missions. Now, there is one current Category 5 restricted mission that's taking place. The other Category 5 restricted missions were, again, Apollo 11, 12, and 14. And 13 would have been a restricted mission, but for obvious reasons, it didn't. That necessarily isn't to mention. Now, a Category 5 restricted mission that's currently on its way is the Mars 2020 Perseverance rover, which is a rover that's going to be, it's really can be also be known as an astrobiological rover because it is searching for signs of, I wouldn't say for life, but it is searching for the right conditions for life, or it's searching for evidence that there could have been life there at one point, and also that the right conditions are there for life. Now, one of the interesting things about the Mars Perseverance rover, or Mars 2020 Perseverance rover, which will be landing on Mars in February of 2021, is that it has a sample collection tube. It has a set of sample collection tubes that will be taking core samples from around wherever it's roving, and we'll be placing these into a deposit and essentially dropping off that deposit to be collected later on. This is a mission that is under proposal that will likely take place sometime in the 2030s to return those samples back to Earth. Now, because these samples are, this is still at least a decade away, potentially longer, we don't actually have the capabilities to process those samples just yet. Once this sample return mission actually takes place, you need a couple of years beforehand to actually start building the facilities, or designing and building the facilities to actually process these samples. Now, it would be, for example, a biosafety level 4 laboratory, which is similar to, for example, there are a couple of biosafety level 4 laboratories throughout the United States and throughout the world. One, for example, is at the CDC. Despite that, it essentially would follow the same process that occurred with the Apollo missions, where the samples would be retrieved, flown to the sample receiving facility, so wherever that biosafety level 4 facility is at, and they would go preliminary examination, characterization, and also checks to ensure that there isn't extant life or current or extinct life that is there, and also ensure that it's not hazardous. Once that takes place in that biosafety level 4 facility, they'd be curated and then eventually sent off to research laboratories, wherever, at whatever university that is proposed to study these. So that's for category 5 restricted. Now this leads me back to the next question, this leads me back to the original question that I started off with, which is, are we ready for the search for life elsewhere? Now this is something that I will say that I believe that we are ready for the search for life elsewhere, and we are necessarily undergoing that search. And I say that because while the risk is incredibly low that something could be returning that could wreak havoc here on Earth, whether that be to us humans or whether that be to plants or other organisms, is the protocols that are in place are stringent, because it's basically better to be safe than sorry. So again, regardless, even if the chances are slim, we still want to make sure. And again, that also includes whether we're returning samples here, or whether we're going to another planet. Again, we're ensuring that anything that can fit, or any cells that can fit on this lens right here of my phone, is spread out throughout a lander that's about the size of a car. Now, the likelihood of returning living microbes is slim, and the likelihood that they can potentially infect humans or other terrestrial organisms is also very slim, and in fact even smaller. Now, there have been discussions that any Martian organisms that would be separated from us by about 4.5 billion years, which on one hand means that we wouldn't have protection from it, such as, for example, with SARS-CoV-2. It's a new virus. We don't have any immunity to it, meaning that we wouldn't have any immunity to whatever Martian virus or Martian bacteria is existing and could potentially affect us. But on the other hand, because we've been separated for so long, it's possible that whatever hypothetical life is on that other world wouldn't necessarily have the capability to make the jump either to us humans or to other plants or to other animals. Now, before I close out, I did want to mention that a lot of this information came from the book When Biospheres Collide by Michael Meltzer. This is a book that is freely available on the NASA website, and if you are interested in planetary protection, I highly recommend reading this book. It's very detailed in terms of its history and in terms of what procedures are taking place, more than I can actually ever cover in a short presentation as I've done here. And I'd also like to thank Dr. Rakesh Mogul, John Rummel, and Ronald Atlas for presentations that either were sent to me or that were made available so I can put together this presentation. And with that, I'd like to thank you for watching, and I hope you stay healthy and stay safe. Thank you.